CIDER, a novel framework, provides causal explanations from subgraphs to labels or phenotypes by training a graph neural network, GNN, solely on high-dimensional measured data. The framework divides the whole graph into two subgraphs: a causal subgraph that affects the label and a spurious subgraph that does not. CIDER directly predicts the distributions of these subgraphs, enabling the estimation of causal strength, i.e., each subgraph's contribution to the graph's label. The framework models the problem as a variant inference task, predicting the two subgraph distributions using a VGAEBASED framework with two channels. The two distributions are then used to reconstruct the original graph structure, and the contribution of each subgraph to the label is computed by performing sample counterfactual implementation in the spurious subgraph. CIDER adopts a diffusion process to enhance the inference capability, where denoising and distilling the spurious subgraph are performed at each diffusion step. After T steps, each step's causal subgraph distribution and noise distribution are obtained. The noise distribution's significant attribute and counterfactual invariance make CIDER trainable, resulting in a sparser and more reliable causal subgraph distribution. CIDER provides not only a causal subgraph due to counterfactual implementation but also its causal links due to the diffusion process. Experimental results on both synthetic and real-world datasets, including single-cell RNA-seq datasets, demonstrate CIDER's advantages over state-of-the-art methods. Theoretical results of CIDER ensure interventional causality, inferring the causal subgraph against an output label or phenotype of a sample based only on data. The conceptual illustration is given in Figure 1. A random graph is defined as G equals X, V, E, where each node has a d-dimensional feature vector, and X is a V times D matrix representing the measured data of a sample or graph. The goal is to derive the conditional distribution of the edges, P, E, X, V, from the data. CIDER, a novel method for identifying and explaining causal relationships within complex graph structures, demonstrates exceptional performance in both synthetic and real-world datasets. In the context of the synthetic BA2 motif dataset, CIDER achieves nearly 100% accuracy in classifying graphs based on attached motifs, underscoring its effectiveness in addressing pattern recognition challenges within graph data. Furthermore, the model outperforms all baselines on real-world datasets such as MUTAG and NCI-1, indicating its ability to learn the causality from the subgraph to underlying phenomena and predict the ground truth using fewer edges. The CIDER method is applied to real-world RNA-seq data analysis, specifically in identifying underlying biological mechanisms related to COVID-19. Using a graph neural network, GNN, a gene network with 370 nodes and 498 edges is constructed, achieving a classification accuracy of 0.85. After applying CIDER, the network is streamlined to 30 edges, maintaining a classification accuracy of 0.82. This result suggests that CIDER preserved approximately 96% of the performance in deducing genogene causal subgraphs while reducing the network's complexity. Validation of CIDER using the COVID-19 RNA-seq dataset from blood reveals that the top 30 genes with the highest causal association scores are partitioned into 3 independent sub-networks, potentially indicating distinct network functions. The isolated COTL1 gene may have a unique impact, given its role in the immune response and involvement in the superpath's innate immune system. Enrichment results from KEGG pathway, gene ontology, and UniProt keywords further support the effectiveness of the CIDER method in identifying COVID-19-related genes, emphasizing their role in inflammatory conditions and potential prognostic value for severe cases. The downregulation of the COTL1 protein in the airway mucus of COVID-19 patients is also noted. Overall, CIDER demonstrates its capability in identifying causal genes and cell-type networks, facilitating the identification of underlying biological mechanisms at a molecular level. The authors applied the CIDER method to identify genes associated with COVID-19 and acute myeloid leukemia, AML, using the TCGAL-AML dataset. This approach led to the selection of 51 key genes with high causal association scores, maintaining an 88.6% accuracy compared to the original gene network. Further biological analysis using tools such as David and String revealed a division of these genes into two distinct clusters, suggesting unique functions and interactions within the molecular network related to AML and ribosome. Enrichment analyses showed strong associations with leukemia, particularly AML, and the pathway in cancer. Two significant sub-networks were identified, one related to AML and the other to ribosomes, suggesting a potential relationship between AML and ribosome dysfunction. Key genes like RPL26, RACK1, and RPS3 were implicated in AML progression, with potential therapeutic strategies targeting these genes. The focal adhesion pathway was also identified as a significant player in AML progression, and targeting this pathway with specific inhibitors has shown promise in preclinical studies. Genes such as KIAA1549L and CPNE8 were found to play critical roles in AML development. KIAA1549L was linked to the development of acute leukemia through its involvement in fusion genes, while CPNE8 negatively regulated AML cell proliferation. The enrichment and biological analyses of key genes from TCGAL-AML data using CIDER provided significant insights into the molecular underpinnings of AML and related leukemias. CIDER, a new method for causal subgraph inference, leverages both counterfactual and diffusion implementations to provide generalizability in evaluating a subgraph's causal impact on labels. It can reduce the influence of unobserved confounders, a major advantage of this work. However, there are remaining issues that need further improvement in this framework, including handling unobserved confounders. CIDER, a novel framework, leverages graph neural networks, GNNs, to identify and explain causal subgraphs in complex systems, enhancing transparency and trust in AI models. The model-agnostic and task-agnostic approach utilizes an additive noise model and counterfactual invariance to infer the causal subgraph by training the GNN on measured data. The framework is based on the concept of random graphs, used to analyze complex networks in various fields. The authors formulate the problem of causality as a causal effect problem, focusing on the causal relationship between the causal subgraph E_C and the label Y. The paper introduces the concept of spurious subgraph E_S, defined as E_C, and formulates the problem using equations 5 and 6. These equations involve sampling a new spurious subgraph E_S from the marginal distribution P, E_S, E_C, without affecting the GNN's output, making the inference from association to causation. The authors adopt an additive noise model, ANM, and a structural causal model to understand the relationship between signals and their noisy observations. ANMs are widely used in various machine learning fields, enabling the identification of the causal subgraph E_C. To identify E_C, the authors assess the causal effect of E_C, requiring intervention in E_S. This is tackled by adopting an ANM, formulated by EQs 7 and 8, where E, Y, and Z represent edges, graph labels, and hidden variables, respectively. The task-agnostic phi can be viewed as message passing, essentially acting as a sequence of transformers based on the edges in the feature matrix X. This perspective allows the problem to be formed as an out-of-distribution, OOD, detection task, aiming to identify a set of edges that can be removed without deviating from the original distribution. The authors use variational inference models to fit the distributions of E_S and E_C, and obtain the subgraph of E that contributes the most to the label Y. Here is the combined summary. The CIDER framework, a two-channel variational graph autoencoder, VGAE, is employed to simultaneously infer causal and spurious subgraphs from gene cell expression matrices. The model operates on the principle of a diffusion process, where the hidden variable Z is treated as a diffusion process, akin to DALLE2. The loss function, LCIDER, consists of three components. L1, which calculates the causal effect of E on Y. LKLD, the KL divergence of ZC and ZS with the normal distribution. And LRE construction, the reconstruction loss of EC union S and E in the form of cross-entropy. The diffusion process in hidden space is characterized by the distributions p, zct, zct plus 1, and p, zst, zst plus 1, which are both normal distributions with parameters micro c theta, ect, sigma c theta, ect, micro s theta, ect, and sigma s theta, ect. The pseudocode for CIDER, outlined in Algorithm 1, demonstrates the process of sampling ZCT and ZST from their respective distributions, updating ECT1 and ST1 using these samples, and then repeating this process until t equals 0. The analysis process on biological data begins with data cleaning on the original gene cell expression matrix, denoted as ZORIGIN, involving removing cells or genes with 0 expression values. The selected gene cell matrix is then used to construct gene and cell type networks, essential for CIDER's analysis. CIDER is employed to generate explanation networks indicating the causal effects of genes and cell types on COVID-19 and influenza. The study draws on various references, including works on graph neural networks, parameterized explainers, and generative causal explanations, which are crucial for interpreting complex models and identifying key factors influencing disease states. The paper also references a blood atlas of COVID-19, protein-protein association networks, functional enrichment analyses, coactose in like proteins, pathcards, and a proteomic profiling study, all of which provide valuable insights into the biological processes involved in the disease and aid in understanding the role of different genes and cell types. The research paper explores the multifaceted relationships between various biomolecules and diseases, including COVID-19, acute myeloid leukemia, and breast cancer. S100 proteins, specifically S100A8 and S100A9, are implicated in the regulation of NADPH oxidase 2 activity during COVID-19 infection, potentially contributing to hyperinflammation. The study also highlights the importance of calcium levels in COVID-19 severity, with low serum calcium levels associated with increased mortality and severity. Furthermore, the paper discusses the potential therapeutic applications of plant lectins, which exhibit antiviral properties against enveloped viruses, and the significance of the complement system and mannose-binding lectin in COVID-19 patients. The relationship between RNA splicing alterations in acute myeloid leukemia and poor prognosis is also emphasized, underscoring the importance of understanding molecular mechanisms underlying cellular stress responses. The paper also explores the connections between breast cancer and acute myeloid leukemia, focusing on the roles of p53 and ribosomal proteins. The high prevalence of p53 pathway dysfunction in acute myeloid leukemia is associated with poor prognosis, while ribosomal proteins, such as nucleolin and RPL26, regulate p53 translation and induction after DNA damage. Additionally, the paper discusses the relationships between RACK1, RPS3, and focal adhesion kinase with proliferation, chemotherapy resistance, and poor prognosis in various leukemia subtypes. The study highlights the complex interplay between genetic factors and their implications for disease development and progression. Novel fusion genes, such as PAX5KIAA1549L and RUNX1C110RF41, are identified in pediatric leukemia subtypes, emphasizing the importance of understanding molecular mechanisms to identify potential therapeutic targets and improve patient outcomes. The research paper cites a wide range of topics in machine learning, graph theory, and network analysis, including graph-based methods for network analysis, semi-supervised classification with graph convolutional networks, and robust counterfactual explanations on graph neural networks. The references also cover asymmetry learning, certified adversarial robustness, and interpretable graph learning via stochastic attention mechanisms. Furthermore, the citations touch on topics such as empirical model approaches for evaluating pollutant diffusion, the role of social networks in information diffusion, and generative modeling by estimating gradients of the data distribution. The performance of the proposed CIDER framework is evaluated on two benchmark datasets, MUTOG and NCI-1, with statistics presented in Table 2. The experiments utilize the atom optimizer with a learning rate of 0.001 and weight decay of 0.0005, with a batch size of 128 and training for 500 epochs. The CIDER framework addresses the limitations of existing graph explainability methods by leveraging a causal inference framework to identify causal relationships between graph nodes and the target outcome. The method involves estimating the causal effect of each node on the target outcome using a diffusion-based approach, which enables the identification of causal and spurious relationships. The experimental results demonstrate the effectiveness of CIDER in identifying causal relationships in graph-structured data, with superior performance compared to baseline models in terms of precision, recall, and F1 score, as shown in Figure 1. The ablation study in Table 3 highlights the significance of the diffusion-based approach and the importance of considering spurious relationships in graph explainability. Overall, the proposed CIDER framework provides a novel approach to graph explainability, addressing the limitations of existing methods and demonstrating superior performance in identifying causal relationships in graph-structured data.